Our world is rapidly becoming an enormous pile of plastic waste. There are six profane large garbage patches bulging in the ocean. Even regions as far separated from us as the Arctic are not safe. We are slowly strangling a lot of ecologies with our trash. Fish, birds, and other animals all unconsciously consume the 5 trillion tons of plastic scattered in the ocean, and doing that is killing them. Scientists are trying to come up with some solutions to solve the plastic pollution crisis, and they're thinking small in a good way. In new research to be published in the Proceedings of the National Academy of Sciences, a team of international scientists explains how they created, by accident, a new enzyme capable of breaking down plastic bottles. This fortunate development could finally enable us to fully recycle plastic drink bottles for the first time in history, causing a much and long-needed dip in plastic pollution increases. The new study tied to the 2016 discovery of a bacteria in a Japanese waste dump that had been evolved to use PET polyethylene terephthalate, utilized in the 1 million soft drink bottles sold every minute around the world, as an energy drink. Team of scientists originally began rolling tests to see how the bacterium, Ideonella sauchinensis, managed to produce an enzyme capable of degrading PET. Those tests, it turns out, accidentally made the enzyme, PETase, even better at degenerating PET. The mutant PETase presently can take only a few days to break down PET, compared to the 450 years it needs for the stuff to degenerate naturally. The salient takeaway is to use PETase to break down plastic bottles before they end up in the environment. However, the enzyme could augment plastics recycling approaches themselves. Current recycling strategies for PET bottles mostly focus on mechanical recycling, so they chop the bottles up and use them for applications that typically do not need the same materials requirements as bottles. Says study co-author Greg Beckham, a researcher at the U.S. Department of Energy's National Renewable Energy Laboratory. Engineered enzymes that break PET down to its building blocks would enable the ability to do full bottle-to-bottle recycling. It would probably help reduce oil drilling demands for new plastic productions. After characterizing the structure of the enzyme by bludgeoning it with X-rays from the UK's Diamond Light source, which can hit objects with laser beams 10 billion times brighter than the sun and help scientists characterize individual atoms from one another. The team changed the structure of PETase a bit to compare it to other enzymes, which breaks down cutin, a polymer found in the protective coating in plants. That process unexpectedly gave PETase with a 20% improvement in degrading PET. This isn't exactly a revolutionary shift from the original, but it does predict the potential to go further and turn the enzyme into something with significant applications. Beckham and his team have already filed a patent to register with the hopes of making the enzyme stable and active at temperatures above 158 degrees Fahrenheit, where PET becomes rubbery and break down 10 to 100 times faster. Akin to laundry detergent, which contains many enzymes that are produced at the industrial scale to clean our clothes. One could envision a PET-degrading enzyme system that is able to take PET bottles in a large industrial reactor with hot water and break the bottles down rapidly to their building block constituents, says Beckham. Many groups around the world are working towards this goal now, and I anticipate that this will be a reality in the next decade or, hopefully, much sooner. Primarily Ideonella sauchinensis bacteria is way far from the primary living species to possess plastic-eating proclivities. Waxworm caterpillars have been found to break down plastic in a matter of hours. And mealworms possess abdomen microbes that eat through polystyrene. Beckham thinks, given how everywhere environmental pollution has become, It is likely that microbes are evolving faster and better strategies to break down man-made plastics. It seems that nature is evolving solutions. Clearly humans could stand to develop some solutions as well.